But on the left is the structure using cryo-EM, the structure of rod outer segment, which contains the visual pigment, rhodopsin in rod cells or cone in cone cells. And this is one of the largest structures done by cryo-EM in collaboration with Wolfgang Baumeister. This is about 1 micrometer cell in diameter, and we're able to obtain those images after a quite long process of learning how to do large structure by cryo-EM. You can see it's loaded with protein. The darker pieces is just a protein. Once you have digital images, you can do whatever you want, and particularly for artistically talented people, they can put the colors, and it shows that the membranes are stacked on top of each other about 600 times, and they hold in the same distance from one another. There are some pillar proteins which keeps them apart. And again, the plasma membrane and the edges of those discs. So you can look on those discs. They can be isolated and studied by atomic force microscopy. And here you have about 1 micrometer in diameter disc membrane, flattened discs that are only made of very few proteins. This again shows you how simple this system is. So eventually you can memorize all of them. And indeed you can do a mouse knockout in GC1, which is guanylate cyclase 1 and 2. They are not essential for that membranes. ATPase, lipase is not essential. This PRCD is not essential. The G protein complex is not essential for the structure. What is only essential for the structure, 2 proteins. One is rhodopsin, and the other is ROM peripherin. And this ROM peripherin makes this ring on the edges, so it only localizes in this part of the disc membrane, and the rest of it is filled with G protein coupled receptor, rhodopsin. 5 millimolar concentration, the highest anywhere in our body, the expression level of this pigment. So it's so high that you can do atomic force microscopy and touch individual molecules. And what you will be surprised is that there are a row of diamonds throughout this membranes. And this is a lower magnification, but then higher magnification, you can recognize individual molecules. So this is again providing the first direct evidence of oligomerization of GPCRs. This is really one of the hottest area of research in the last 10 years, and finally came down and cooled down. Now I think there is very few who will still believe that GPCRs are monomeric. They really act in oligomeric structure for many reasons. And as a professor of pharmacology, I can tell you why, but this will take another hour. It's also a pharmacological reason by which they must come together in a cluster of those receptors. So, how many of these GPCRs? It's all together in our genome, it's about 800 GPCRs. And rhodopsin, obviously, is the most important molecule in the universe. This one I hope you know. And there are also cone pigments on the same branch. And then we have the rest of those. And they, the largest subfamily is called subfamily A. They are like rhodopsin molecules. So like Ronald Reagan says, once you see one tree, you have seen forest. Once you know the structure of this molecule, then you know exactly the entire family. And that's exactly what happened in the last 5 years or so. 24 different GPCRs, as depicted on this slide, were crystallized, and the structure turns out to be very similar to rhodopsin. And this is a movie made by Sławek Filipek, who is here in chemistry. We have collaborated with Sławek for many, many years. Just recently, I did a very nice study on the structure of water molecule, which are important for activation process. And for some reason, this movie, oh, it's coming. It's coming through this beautiful molecule, which is made of 7 helices, transmembrane helices, and boundary of the membrane are shown here, and going from left from the blue, this will be helix 1, going to red will be helix 7, and on the top cytoplasmic surface. And within this molecule is the chromophore, 11-cis retinal, linked via protonated shift base. And this is very important because this protonation of the shift base determines the wavelength of absorption. I will come back to that in a minute. So that is the chromophore now bent in this 11 cis configuration, and upon activation it changes it to all trans configuration, triggering the activation. So we approach to this by crystallography. Rhodopsin has been first GPCR to be crystallized later on, and it took us only 3 years, and few people working on that were able to obtain light-activated crystal structure of rhodopsin. Doesn't remind you this picture what I showed you with Bill that had a red retina and yellow retina? It is exactly what we're doing now, but on a molecular level, to understanding how this molecule works. Indeed, rhodopsin doesn't switch from one conformation to another, but undergoes conformational changes, and my lab and several others contributed structural information on different intermediates. The intermediate called meta-2 rhodopsin is the signaling intermediate which interacts this conformation, couples to G protein called transducin, and to close the cycle. So the conformational changes to this very fast progressing beta-lumi rhodopsin, meta-1 rhodopsin, it really doesn't change much. The photoisomerization is a femtosecond process, so it's extremely fast, obviously, as you would imagine, that the photochemical reaction can be on that level. It's one of the fastest photochemical reactions in our biology altogether, yet the protein required time before it changes. So the all intermediates you can really superimpose on each other because there is not much of conformational change. The conformational change happens in milliseconds as the rhodopsin undergoes conformational change from meta-1 to meta-2. Now, there is a caveat here because we have chromophore located in this part of the molecule, and we know that the changes, significant, few angstrom changes, so it's not like blossoming of flower. Those are the really protonation and some movement of helices happen in two regions, NPXXY region and dry region. So those are highly conserved in the structure of all of the GPCRs, particularly from family A. So somehow that signal has to be transmitted throughout the entire molecule. That's quite remarkable. How would you do that without large conformational change? That you go from the site of the isomerization to the cytoplasmic surface. Now, if you look on this other structure of 24 GPCRs, they fold exactly like rhodopsin, and yet do not have any sequence homology. So some think in thermodynamics is that those helices are the most stable when they bundle and maybe somehow involved in signaling to this multi-membrane properties. So we have been pursuing a different concept. If you don't have a protonation of those residues as a result of large conformational changes, maybe water molecules is responsible for that. And here we have a footprinting experiment where we use X-ray in synchrotron facility that we can ionize water molecule to lead to modification of specific residues and we can determine the rate of those residues undergoing modification as a result of lysis of water, breaking up, making hydroxyl radical that can react with any residue in the molecule. And then we go to meta-2 rhodopsin again, we can determine the rate, we can color code them, and then finally we go to the opsin molecule, which doesn't have a chromophore. So water is really the signaling molecule that changes its conformation, leading to activation of the cytoplasmic surface. And Swatek just recently published Nature Communication paper in collaboration with my lab to look on those water computationally. And indeed, there's a channel throughout the molecule that transmits that signal from the heart of the molecule up to the cytoplasmic surface. So is the chromophore of rhodopsin the same in color pigment? Because when we look on the colors, we can discriminate minute details of the different combination of blue, red, and green pigment altogether. And not surprisingly for chemists, is that it is universal chromophore. 11 cis retinal is identical in conv and rho, but yet give the rise to different lambda maximum. So in one case, absorb around 420 nanometers. And then on the other extreme would be red pigment, which absorbs 640 nanometers or so. So how this can be done? And there is a clever fellow who lives very close to my place in Toledo, who did the following experiment. He took cellular retinol binding protein, modified in a way that now he can attach the chromophore via protonated shift base or deprotonated shift base. In his case, he used all-trans retinal because, again, the resonance structure of all-trans or 11-cis retinal will be the same. And then he modified many residues around the binding site. And he came up that this same chromophore can absorb 400 nanometers, or 500 nanometers, or 650 or so nanometers. So he can make a color with this particular molecule, all-trans retinal. So interaction of all-trans retinal or 11-cis retinal with the protein moiety will lead to a different lambda maximum. And it's accomplished via this distance between counterion of protonated shift base and some interaction with hydrophilic residues within the binding site. So with this background, what we would like to do is to look at the retina again. You have fovea, which contains mostly cones. And on the periphery, we have rods. So all of the diseases of cone will really eliminate of central vision, but nobody will go blind from that because there will be a residual vision on the periphery. And then rods also could go undergo degenerative processes where the vision is lost slowly from the periphery to a tunnel vision and eventually to complete blindness at the end of this process. So when we plot from the optic nerve the concentration of rod and cones, the picture shows the distribution of rods and cones. You can see on the periphery there are very few cones, and then in the center we have those cones present. And that central part of the retina, it's about 20,000 cells. And there are only 20,000 postmitotic neurons that are with us throughout our life. There is this phagocytotic process of renewal, but yet this cell has to survive such a long time for, again, providing a sensation of vision. Now, this is a visual spectrum, and our perception of light is in this region of visible spectrum. But very recently, my wife discovered there is also infrared vision, and this paper will be in 2 or 3 weeks from now in PNAS. We can detect also infrared vision via a very special mechanism that I encourage you to take a closer look. But today what we wanted to show is the spectrum of this short wavelength, rhodopsin, green, and red pigment as it's in human. And we can derive the structure from rhodopsin, which is not very accurate but close, that we can again position the chromophore very snugly here in the binding site with different interaction with the negatively charged residues or lack of those interactions that give the rise to color vision. And we're really pursuing this right now to crystallize all of the three pigments to have a more precise information about this color spectral tuning. Now I will change gear to another step in vision, is this regeneration. The right side, you remember Kine experiment when he took the retina and then get red again? That's the regeneration and retinoid isomerase. So here we have the molecular map of events as it happened in the outer segment and RPE. From rhodopsin, rhodopsin gets isomerized to all trans and then loses its chromophore. The chromophore gets reduced and it gets retinol transported to the RPE, where it becomes esterified with fatty acid and form lipid droplets like in stellar cells where we have accumulation of retinol for storage purposes. The same happens in RPE cells, and that is because of enzyme called LRAT, lecithin retinol acyl transferase, transfer from lecithin fatty acid and make the carboxylic ester